Okay, great.
So it is my pleasure to introduce Rachel Goodman from Hampden-Sydney College, who's going to be sharing a really cool amphibian pathogen surveillance program that she's been running for many years and some of the data they have with that. Thank you so much.
Hi there. So yeah, today I'm going to be talking about, in my lab, I've actually been doing various disease surveillance work for, I guess, about 16 years out at Hampden-Sydney College. But this is a more recent effort that I've started up with a collaborator, Amanda Starr at Longwood University, which is also in Farmville, not too far from here. Oop, and I clicked the wrong thing. Here we go. I got it. So I'm sure this crowd is very aware of the current threats of amphibian declines. We know that all wildlife is in trouble, but there are estimates that something like 41% of named species are in some category of threat or endangerment. And so habitat loss, destruction, climate change, these are really important, but disease is also a huge threat to amphibians. And of course, most amphibians are not facing these threats in isolation, right? So these are piled on top of each other. In my lab, we've worked a lot with ranavirus, which I won't be speaking about today. But for this project, BD and B-Sal are the pathogens which I'll be introducing that we know have caused significant mass mortality events and declines in amphibians. If you're not familiar with BD fungus, Batrachochytrium dendrobatidis, it is a fungus that infects certain kinds of skin in amphibians, the kind that contains keratin. And while it can infect various amphibians, it's mostly a problem for frogs. And when it causes disease, it's called chytridiomycosis. So it does not cause disease in all species or life stages, but when it does, it's essentially completely disrupting the skin function, which we know is important for respiration, for osmotic balance, and can eventually lead to heart attack in the frogs, as well as the skin sort of sloughing off over time. And so this can be spread frog-to-frog contact, but also through the water, through the environment, and through researchers or people touching frogs and then touching other things or soil that's being carried around on our boots. So as I mentioned, it does not impact all frogs, but it has been attributed to causing amphibian declines in at least, well, over 500 species, and potentially 90 extinctions have been caused by this pathogen. So it hasn't really been that impactful on salamanders generally. Most of you have probably seen a documentary with frogs and the BD fungus. It is very devastating. But here in North America, our frogs have not, they've handled it a little better. The second pathogen I want to talk about is B-Sal, which is a related fungus, but it's called Salamandra vorans for like voracious, right? Because it eats salamander skin. And so both of these fungi originated in Asia and then have spread throughout the world. And this B-Sal fungus just showed up in Europe in the 2010s and has been causing some devastating major declines in some salamanders there. So again, it's consuming the skin, and it looks a little different because in salamanders, oops, I'm missing a picture. Oh, there it is. It actually causes these like ulcerations that are really notable. And again, it can be spread salamander, or water, or environment, or people, right? And it's a huge concern for those of us in North America because we have the highest salamander diversity in the world here, and especially very nearby in the Appalachian Mountains. That's the figure on the top showing that red and yellow is where most salamanders are in the world. And if this B-Sal fungus gets to North America, we think it could be really devastating. So one of my former students was involved in a group of studies where they tested in a laboratory, very secure facility, they tested 35 North American species, found that 71% of them will get the fungus, will get sick, or will become infected. And a third of the species die within 3 weeks, or in about 3 weeks. So we can only imagine if this were released into the wild. And so we're very concerned about knowing when this pathogen shows up, and then I guess everyone descending to try to eradicate it. So SNAPS is the Student Network for Amphibian Pathogen Surveillance. And the idea here is to recruit undergraduates in classes across North America to continuously monitor. So we've seen with a lot of citizen science projects that there is sometimes a challenge to keep people collecting the data year after year. And so the idea here is to have students do it nested within courses, and SNAPS provides materials, training, educational units, and the supplies as well. And so it was founded in 2020. We got involved a few years ago, and you can see there that there are many institutions, many students, many samples have been taken. So we joined in 23, and so we've just been doing this for a few rounds, and we're doing this in different courses to be able to keep doing the sampling. And so we have a handful of samples shown here. In 2024, we're trying to get more salamanders. I have not worked as much with salamanders, so I'm not as cued into finding them on campus. And then we're planning to do it again in the coming spring. And so we catch the salamanders and frogs. As was mentioned, it was the Rockwood Nature Center, which now I'm really excited to probably take students on a field trip and learn more about it. But as they mentioned, biosecurity is very important. So all the animals are handled separately, kept in separate bags. I mentioned we do have a lot of air inside the bags. When we're measuring, it looks like there's not very much air for that moment because we're just sort of holding the salamander in place. But we're measuring them. And then importantly, we're taking skin samples with swabbing across the skin. I'll show you what the protocol is on the next slide. And returning them to their capture location. Then all of those swabs are stored in a freezer, and they're sent to the SNAPS program where they do the DNA testing using qPCR, which is sort of like when you would get your COVID test back in the day. Or I guess you still could get it, but now we have the home kits, right? When you would actually send in the DNA tests. So there's a standardized protocol. So again, they provide videos and instruction on this if people haven't done it before. And it's a certain number of swabs taken in a certain way across certain sites. So you're essentially going back and forth either 30 or 35 times, depending on the animal. Oops. And again, I was so happy to see someone else talking about biosecurity. We're always wearing gloves in all these pictures. There's actually a turtle trapping picture up there, but from other research. But we're always using gloves. We're always using disposable containers, or if they're reusable, like Tupperwares, they're washed, rinsed, disinfected. And there are standard protocols for this. So it's not just like, oh, you just scrub it out with some Dawn or something. There are certain chemicals that you can use that are approved for like BD, B-Sal, ranavirus, et cetera. And then whenever we move, I'm generally working now just at Hampden-Sydney, but when I start working in other places, other countries, I'm always very careful to thoroughly wash and disinfect all of my gear and all of my shoes before I go somewhere else. So the good news is that we have not found any B-Sal, and no one has, to my knowledge, in North America. Hopefully that will continue to be the case. It's important that we continue monitoring. As far as BD, we had seen it before on campus, so I know that it's, and as have people in other areas regionally. But these are our results so far with respect to BD. And I apologize for having a lot of Latin names up there. I was on my slide trying to make room for everything. But we had one BD positive southern two-line salamander. And then among our frogs, we had 3 BD positive bullfrogs and, oh, I'm sorry, three pickerel frogs positive and 1 bullfrog. So the other frogs that we have up there where we have not seen any positives, that would be cricket frogs, spring peepers, green frogs, and Fowler's toads, where we have not seen BD positives. That's only 2 seasons though, or 2 samples. For our last one, we sent the samples in, but then there are no results because of the government shutdown. So the lab for, I'm not sure if it's the entire program, but where I think most of the samples are sent and processed is it's under the umbrella of USGS. And so when you send an email, it says, sorry, we are not able to do email. So as far as the rates of positives that we see, again, in the fall sample, about 10% were positive, and in the spring sample, 7% were positive. And just as a point of comparison, in 2010, I did another study with students just in the Hampden-Sydney area. And at that time, and now I have common names for pretty pictures, although it'll be really fast for you to look at that time, 7% were positive. So it seems to be in line with what we've seen previously, although slightly different composition of the frogs that were carrying the BD. And so cricket frogs and spring peepers in this case were carrying it. So it looks like most of the species that we've collected, with the exception of the Fowler's toad, I guess we have not seen BD in. So as far as challenges, so I think this program is wonderful, and it's so important that we keep monitoring for when BSAL, well, I say when, hopefully if BSAL shows up and it's a no, but the issue is having that continued sampling. And while the stuff we do out in the field is very cheap and relatively easy to do, the lab work is expensive. Doing quantitative PCR, which is a method of duplicating the DNA and seeing how much there is in the sample, it's fairly expensive. And so right now, I mentioned the government shutdown, but also the National Wildlife Health Center, which is where the people have been running all these DNA tests, they've informed us that there's a potential for their budget to be zeroed out, apparently. So I don't know if that is a definite or not yet. Even before the government shutdown, we were being told you have to get your samples in by the end of September, and then we can run them in October, because after that, potentially, I don't know what's going to happen. So I'm a little worried, but I also don't know the details. I'm not in the mix that much. It may be the case that other sponsors financially will have to pick up the shortfall, and then, you know, I don't know what it means in terms of where the DNA tests will be taking place. So as for myself and my collaborator, we're going to keep collecting, and we totally have the ability to test for BD and BSAL. I don't have the reagents on hand right now, but if it comes to pass that SNAPs can't do that anymore, then we will commit to continue doing that. And my hope is that maybe through this network, others who have the ability could do that, or even, you know, for example, once we're set up, we could maybe take on testing from other places if we had a funding source. But we're also interested in other, in terms of our labs generally, we're also looking at other pathogens. So you may be familiar with ranavirus and Purkinje as important amphibian pathogens. And then my collaborator, Amanda Starr, works with, she's more of a microbiologist. And so we're going to be doing microbiome studies where we essentially sequence the DNA of all the bacteria in the skin and see how that interacts with the 4 pathogens that we're looking at potentially. But that will be a subset of samples, because that is even more expensive to do, as you might imagine. So with that, I would just like to thank, of course, all of our enthusiastic students who have been involved, and these other groups. And then I don't know if there's any time for questions.
There probably is. And in fact, we actually had a really good question in chat that I'm going to just start with, which is what chemical can you use at home for disinfection of reusable containers that the residue is not toxic to the animals?
That's a great question. So if you go on to, it's going to depend on what you have and what the concentration is. So for example, even just saying, you know, bleach, well, what's the residue going to be? But even then, what percentage of bleach and how many minutes? Those things are very specific. There are other, I don't use that. It's also highly corrosive to all your stuff. I use Virkon, which is, I think, pretty widely available. But again, the percentages and the times on those, if you go online and you look, I would say the best thing is to look in PARC, P-A-R-K, has a whole disease task force. So, sorry, K, P-A-R-C, Partners in Amphibian and Reptile Conservation, has a great disease task team, and they have specific where you can like look at the different possibilities and see what's most affordable. And also in terms of traveling. So I have learned, you know, traveling with toxic chemicals is difficult. So I like Virkon because it's a powder and I can buy little packets and take it to other countries without getting in trouble. So
we can do one more question. Yeah.
Scale, I'm just curious.
yeah, there definitely are. I think in all cases it's an issue of funding for the long term. So, you know, one of the, you know, this is focused a lot on undergraduate institutions and, you know, then this USGS agency, one of the things with having like an R1 lab host this is that R1, you know, research labs are constantly have the pressure of publication and new grants and new grants and new publications. And so with this, you're signing on to doing this long-term monitoring and you may not find it. And we know BD's out there and this, you know, so it's like, if you do this for 5 years and you get hardly any, you know, publishable new exciting stuff, that's not, so it has to be through some sort of network of, you know, labs or organizations or a single institution, perhaps, but one that's dedicated to the importance of the long-term monitoring that's not under the pressure to, you know, produce public, you know, publications in that same sense. So yeah, I'm not sure. And I'm not like at the level, I'm kind of just out on the, you know, the little nodes of this. So probably in the interior, I'm sure there are big discussions right now about what happens, especially if that, you know, budget's going away, like where this testing can be rerouted to.
Okay, and we actually have a question in the forum, if you don't mind taking a seat and answering for that, and then we can move on to the next presentation, which I'll get queued up here for us.